Hello, my name is Sabine Bahn. I work at the University of Cambridge and also the Erasmus Medical Center in Rotterdam. And my talk is about developing a blood test for schizophrenia. My French is unfortunately very limited, so it's a great pleasure to be here. Thank you so much. And please interrupt me if you have any questions as well. And I try to speak a bit more slowly than I usually speak. I'm going to talk to you and tell you a little bit about what we've been doing over the last sort of 10 years. We've been trying to develop a blood test to diagnose or aid in the diagnosis of schizophrenia. Now, this is quite a controversial thing for many psychiatrists. We don't think about psychiatric disorders of being systemic disorders, but I hope that at the end of this talk you will at least sort of consider that this may be the case. So let me try to convince you. So we started off with our research about, 10, 15 years ago, and we started off with post-mortem brain, because for me this is the only substrate where we may identify something which has to do with the disorders. We haven't got animal models at the moment which really truly reflect the pathology of schizophrenia and other psychiatric disorders, because we don't know the etiology as yet. I hope you all agree with that. The post-mortem brain, however, has a lot of limitations. People who died with a diagnosis of schizophrenia often were ill for tens of years. They were in psychiatric hospitals. They ate differently. They were exposed to different environmental factors, pathogens, etc., etc. So they will not tell us the whole story because it's an end stage. However, it gives us some idea. And what we found in post-mortem brain is very prominent changes in mitochondrial function, in glucoregulation, and other sort of much more fundamental biochemical abnormalities. It also implied that maybe in schizophrenia at some stage during the disease there are some hypoxic processes which lead to abnormality in oxidative stress mechanisms. So this was quite an interesting observation, but we had to really trace it back to to confirm it really in living patients. So this is when we moved on to cerebrospinal fluid. And there we collaborate with the University of Cologne, and they have several hundred cerebrospinal fluid samples from first episode never drug-treated schizophrenia patients who come in with their first presentation. They have a lumbar puncture and also serum collection. And we did these studies, and there we also found changes in glucose, in lactate and other metabolites, which implied maybe there is a problem in this glucoregulation in the brain of schizophrenia patients. I'm not going to go into the details. This has been published. If you're interested, you can read about it. But this was the first time that we see that changes and markers which we find in the brain, in the post-mortem brain, are also there in living patients, first episode patient in cerebrospinal fluid. And this is when I had the idea, maybe it's also systemic, maybe the changes that are in the cerebrospinal fluid are also in the serum. And that could well be the case, because per day I think half a liter of cerebrospinal fluid is being synthesized, and it's all being eluted into the blood system, into the venous system. So it could be either markers coming from the brain, going into the CSF and then into the bloodstream, but it could also be that maybe schizophrenia is a systemic disorder, like diabetes and so forth, where the pituitary is affected in the peripheral system, etc. as well. So in the brain they may have slightly different function in the periphery, but if there are abnormalities, the peripheral changes may track the abnormalities in the brain. They may just be a marker as well of the disease. They may not cause the same problem in the periphery as they do in the brain, particularly also as the brain and neurons are very specific in that they're post-mitotic, in that they can only use glucose and not other metabolites as energy transferring sort of metabolites. They can't use lipids, for example, very effectively. So the brain is different in a different way. And maybe this is causing these abnormalities. So this is really what led up to the sort of next stages. On top of that, we have an armory of different technologies in the lab in Cambridge. And in Holland, I have a lab in Holland as well, in Rotterdam at Erasmus Medical Center. So we started off with looking at gene expression a lot. But over time, over the years, we found that very little changes can be seen at the transcript level, at the expressed genes. And even in an animal model where you do something quite severe, the brain does not seem to change the transcriptional machinery very much, even under severe conditions. So we moved on to proteomics because the proteins are really the effector proteins, the effector molecules. So we have the genes, we have the transcripts, but only the proteins are really driving the mechanism, the enzymes, etc., and the metabolites downstream are associated then with the disease. And we found also when we looked at the gene expression that many changes in gene expression, for example, in brain, are not translated into proteins. It's only about 20%, so it doesn't really mean very much. It is some sort of transcriptional dysregulation, but it will not have downstream effects because it's not translated into proteins and into functional molecules. So we're using mass spectrometry in the lab, but also immunoplexed ELISAs, and I'm going to tell you a little bit about these techniques later on. We're now also looking at phosphorylation because it's not only the protein expression, but it's also the activation of the proteins via phosphorylation of enzymes, which make them active, and the dephosphorylation deactivates them, which is very, very important in the brain. So because a lot of the effects have to be very rapid of neurotransmission, so it doesn't involve proteins or genes being expressed and proteins being translated, but it just is quite a sort of fast switch between phosphorylation off and on, basically switching enzymes on and off. So this is something which we're getting very interested in. So we're looking at post-mortem samples at living patients and also at animal models of psychiatric disorders. I will not talk about the animal models today. I will focus on the development of a blood test to aid in the diagnosis of schizophrenia because I think this is the most important result we have. It's also the most clinically relevant because this is really what I hope to do, is to bring something to the clinic. We can try to understand all sorts of complicated things in the brain, have hypotheses, but if it's not clinically relevant and leading to better drugs or better diagnosis, I think I'm not that interested in it because I'm also a practicing psychiatrist, so I really want to see something in the clinic. So if we had biomarkers in the blood, which is easily accessible, which relate to the disease diagnosis, but also to different processes here, like risk prediction, can we identify, and we're going to do a study on prodromal patients. This is with Mario Del Krebs here in Paris as well. She has collected prodromal schizophrenia ultra-high risk patients and followed them up into transition or not transition into schizophrenia. And we want to see, can we predict before someone gets ill whether they're going to have these disorders? So we can also then patient stratification. Already we see that patients have, schizophrenia patients, about 50% have metabolic alterations in insulin function. We know that schizophrenia is associated with diabetes, and that has been known for the last 50 years, well before the antipsychotic era. But also we see a lot of inflammation markers. So are there different subgroups of patients, and is our success rate of treating patients so poor because we just treat them with a standard drug and we don't subtype them? If syphilis, neurosyphilis, will still not be identified as such, the pathogen wouldn't be identified, we still would also call them schizophrenia patients, probably treat them with the same drugs, but obviously not with too much success. And I think a lot of studies, including Marion's studies, are looking into these environmental factors. The other thing is to predict drug response, and we have some encouraging results in depression where we have patients who have been off medication or are drug naive, and we take blood and we follow them up. And we can actually quite well predict those patients who will easily respond to medication on the basis of a number of biomarkers. And these things, these tools, these personalized medicine approaches hopefully will enter also into psychiatry at some stage. They have entered into the field of the cancer field. We know all about the breast cancer staging and the respective treatment according to the profile of a patient. So these, I think, may be things in the future which will enter into psychiatry as well. Thank you very much. So if we just put the whole thing on. So our aim was, because we had a lot of samples from first episode never drug-treated schizophrenia patient, to see, can we identify schizophrenia patient at this very early stage in their serum signature and separate them from control patients. Obviously, there are the other indications, in particular the prediction of whether someone from a prodrome is going to convert, which is probably even more important. But this was something where we had quite a few hundred samples, and we started to work with that quite extensively. And because I was interested to bring this to the market, I set up a company, Sinova Neurotech, and we have some MBA students in my department as well, and they did some market research. So we had to, before we went to investors, say, how would that help, such an early diagnostic test? And they looked at the literature, and as you can see, you know, there could be quite dramatic implications of early diagnosis, because we all know patients hover around with mild psychotic symptoms. They may not be admitting to their hallucinations, delusions. So they may be ill for months, if not years, before they get properly diagnosed and treated. In the meantime, their life is being severely affected. They may drop out of education. They may get involved with drugs, with crime. They fall out with their parents, etc. So their life is very much negatively influenced in a very profound way, which you can't repair afterwards. So an early diagnosis, I believe, is very important. So indeed, if our test, you know, sort of would be implemented to have an early diagnosis of schizophrenia, then we could reduce premature deaths by about 4%, hospitalizations up to 25%, inpatient days by up to 64%. Relapses could be prevented as well by up to 70%. Time to remission could be reduced. Poor outcomes by up to a third. And duration of untreated psychosis by up to 88 weeks. So this would have very advantageous financial as well as clinical implications. So this could warrant to develop such a test, which obviously would cost some money, and so this was really the question, because to develop a blood test costs about 25 million. So you need to convince someone to put that money into the pot, basically, to take it forward. So they only do that if they get some returns. So there was a potential here. They could see that there could be returns. Here also, this is a bit more speculative about prodromal patients, but we could probably reduce the incidence of schizophrenia if we identified patients, ultra-high-risk patients or prodromal patients. Because we know for identical twins, the concordance rate, well, is between 30 and 80% according what you read. But if you look at the Gottesman figures, some of them were even sort of 10-15% in the meta-analysis, which led to the figure of 50%. Which means that although people have a strong genetic predisposition, they're not destined to get the illness. It's only it's only a small percentage actually. So this makes me very hopeful, which means those who get the illness, something must happen to them, some environmental factor, which may be after birth, which may be infections, fevers, illicit drug use, but something is driving the illness to become manifest. So if we know that someone has a high risk, something simple like psychoeducation to tell a young person you shouldn't be using sort of stimulant drugs or cannabis, etc., giving them maybe education as well, how to deal with stress, may prevent it. And I think several studies from Australia, from Pat McGorry, so are implying already that such approaches could be useful. A more, rigorous approach would be to give some medication as well early on. This may be warranted, to avoid a clinical outcome which is not very favorable. Particularly, I think in schizophrenia, you probably have a window between sort of 15, 18 to 25. You probably don't have to treat them forever, but just during this vulnerable period where a lot of pruning and maturation, and also the sex hormone sort of influence probably drive the higher risk of actually presenting with manifest psychosis. So it could be time limited as well, but I think this is something which one could consider. So the techniques that we're using is here mass spectrometry. Again, I'm not going to go into the details. It's basically we take serum plasma, we extract the proteins, we deplete the most abundant protein, which is immunoglobulins and haptoglobin and transferrin, etc., etc. There are sort of 6 of them, which make up 95% of the protein content of the serum. So if we leave that in, we can't see all the less abundant proteins. So we have to pull them out. This is on a column, basically, where your antibodies and this stuff is being pulled out. And then these proteins are being digested. And this generates candidates, but it can't be validated in a very large scale on that system. And this is why we move into antibodies. So we look, are there antibodies available? And in our case, because we had the company and we partnered with a company in Texas, which has a multiplexed ELISA platform, they actually incorporate our candidate biomarkers into their platform, a multiplexed ELISA platform, which I'm going to tell you about next. This is a very clever system and is quite unique. So it's based on the Luminex platform. You may have heard about that. It's so the people who set up the company Luminex, then they split up and they developed into a diagnostic arm, which is the Texan company who have well, basically acquired at the time 80% of my company, and we partnered together quite closely. So this allowed us to transfer these assays onto this platform. So you have hundreds of beads of different colors, and you coat them with a specific antibody, and you know color so-and-so has the antibody against the apolipoprotein B or against interleukin 6, etc., etc. And then you have a secondary antibody which is fluorescently labeled, and then it runs through a fluorescent activated cell sorting system, the beads, and then you can quantify with absolute quantitation how much of this molecule of this specific analyte is present in your sample. And it's very clever because we can put together up to 250, well, we have at the moment 260 analytes, human serum analytes, which can be measured simultaneously, basically, not in the same reaction, they're all in 6 or 7 plexes. But it means that you only need 500 microliters to measure so many proteins. If some of you may have done sort of normal ELISAs, enzyme-linked immunosorbent assays, there you need already 300, 400, 500 microliter for one analyte. So if you wanted to measure 500, you would need to take a liter of blood from a patient, which they clearly wouldn't be very happy. So this is what we call a disruptive technology. This is a technology which allows you to do something differently now because it's the scale. You don't need as much serum. Because we assume that there's not one marker for schizophrenia or depression. It will be a panel of markers. And it's a combinatorial, so we need the statistics to put something in which gives you a probability which is related to molecule changes. And this becomes reliant really on such technology, which is much more high throughput, which is precise, reproducible, etc., etc. And this wasn't the case in the past. And at the moment, if we just click another, we have, well, this is not the latest one, but we have several hundred, I think it's now 7, keep going. Yeah, I think it's now 600 or 650 assays which are available. And they relate to key metabolic pathways, in particular inflammation, metabolic cytokines, cancer mark growth, etc. You can read it here. But the nice thing for our rodent study is also that 90 assays from the human panel are also in rodents. So we can do serum study, translational studies in these so-called animal models of psychiatric disorders. Again, I haven't got time to talk about it, but this gives a good opportunity because these things are often missing in psychiatry and neurology. This sort of translation, because we can't take out bits of brain, you know, like people in the cancer field at different stages. But if we have a serum correlate, then, you know, this is giving us an indication of the disease state, etc., if this is closely correlated. And also allows us to take that into the animal models. And this could be very helpful for improved drug discovery approaches, because at the moment we have these animal models who are, when you puff them a little bit of air, you know, sort of it's the pre-pulse inhibition. All these things are not very sophisticated and not really giving you a diagnosis of schizophrenia. But at the molecular level, if we can see that drugs are changing molecules which are associated with the disease, that may be more advantageous and more meaningful as well. So, if we just click further, yeah, and further, yes, and one more, I think, no, one back, sorry. So this was the pilot study. So the company, they saw our work and they got quite interested. And we had samples, schizophrenia samples, about 50 to 100, first episode never drug-treated schizophrenia samples from 3 independent clinical centers from Germany and matched controls as well. So we shipped them to them and they ran them. At the time they had about 150 or so unlights, 149, which we measured. And we found 30 markers which separated very clearly controls from schizophrenia patients. And this is when they put in their investment as well to take that further to the next stage. What was particularly interesting as well, we could predict the schizophrenia patients in Center 2 and 3 based on the results which we obtained in Center 1. So it's not just putting them all together and doing the same thing, but it's an unsupervised examination. So first of all, we look in Center 1, what molecules separate schizophrenia? We found 34, something like that. And then we used the 34 and asked these 34, can they predict whether someone is schizophrenic in these other cohorts from different clinical centers? And yes, that worked quite well here with a very good sensitivity specificity, as you can see. And also we found that the schizophrenia patients were quite different from bipolar patients and also from depression patients. This was a pilot study, but this was very encouraging. And this is when we went to the next stage. And this is just a summary slide. It shows you some of the markers that come up again and again and again, not all of them. And here we show 6 independent clinical centers. Several have several hundred schizophrenia patients in them, of schizophrenia cohorts and matched controls. Number 6 is particularly interesting because it's a cohort from the US military. So the US military has a repository of 45 million serum samples which are collected, and this is through the collaboration with Bob Yolkin, a friend of ours in Johns Hopkins University. And they collect every year or every 2 years samples from all the personnel in the US military to look for HIV and other sort of effects. I don't know exactly what they're looking, and they're freezing them down. And we were able to get samples from patients who, before they actually got schizophrenia, who were later then retired with a diagnosis of schizophrenia. So the patients that we investigated here already show some of the markers of schizophrenia were about 4 weeks before they had an index assessment by psychiatrists, usually for anxiety or something else, which later on turned out to be something more sinister. So this is this was quite interesting, but we see markers, but the markers are not necessarily going all up or down in schizophrenia. So sometimes they just show a change. So we can't always say, well, cortisol usually is increased, apolipoprotein H is increased, but some of them, they may go in certain patients up or down as well. So this again requires then a more complex algorithm because you weight these analyte changes rather than them going up and down. This becomes then quite complicated. We also compared the markers here with bipolar disorder, again also from the US military, pre-symptomatic patients, first episode MDD as well, manic psychosis, which would be quite relevant, whether they actually showed similar markers, and also Asperger syndrome, which we had analyzed as well. So we find quite a consistent signature, and as you can see, lots of the changes, and these are just a few of them, quite consistently measured ones, but we see a number of changes which relate to certain pathways which are in the next slide, hopefully again. It's all my people, I use their slides and they all like these animated things and I don't know how to switch it off. So these are really, if we slide one more, these are the markers we see changing in schizophrenia, very prominent changes in metabolic markers. And I'm going to talk in the next part of my talk about that. Really, many changes in insulin, pro-insulin, growth hormone, very, very consistent, and about 50% of patients. Then we see hormonal changes, HPA axis changes, testosterone levels, sex hormone binding globulin comes up again and again, also in major depression, for example, immunological changes, particularly I think monocytic sort of markers, although it's not very clear, it's not always pro-inflammatory, it's pro-inflammatory and anti-inflammatory, so it seems there's something going on, but the body's actually reacting towards it, so it's not 100% sort of clear-cut story, and this really requires further investigation. Neurotrophic factors, BDNF, other factors, PDGF and things, we're seeing those as well, VEGF, etc. So, next slide, please. Yeah, if you go one up. Yeah, so we had these findings. We had the 34 markers. Initially, we found in these 3 different centers. We did then the further studies on 6 clinical centers. I showed you some of the markers. We looked at bipolar disorder, depression, and eventually we selected 51 markers, which we put into a shrink-wrapped, as we call it, assay system, which means we pulled them out of the still 250 or 190 or how much they were at the time, and then they're put into a different work system. So it's a liquid handling system, so it's all basically robots are managing the serum and measuring them. So they're not in the whole big thing anymore, they're taken out and shrink-wrapped. So we have now a schizophrenia assay system, sort of separated out and validated. So this is because we were going to take this test to the market as well. So we had 809 subjects here. You can see here about half of the 570 patients were first onset drug naive, but we also looked at recent onset and drug treated patients. Later on, we also looked at chronic patients. And again, I will not show you all the details, but we see probably state and trait markers. Again, in the next part of my talk, I will come a little more into that. And we also had matched controls. And we collaborated with Princeton University. We needed quite expert sort of mathematicians to work on this project. We collaborated with Vladimir Vapnik, who's the father of machine learning, which is a sort of Bayesian approach of developing algorithms. He is very famous, and we worked with his son-in-law, who's another mathematician, Rauf Ismailov, and they helped us to develop an algorithm based on these samples and investigation, which gives us a very good sensitivity and specificity. And in the next slide, this is just outlining you this algorithm, how good it is. So we have here the green ones, which is the control patients who have been accurately identified, and then the blue ones is schizophrenia patients. And on the SVM support vector machine A is really done on the samples which were run in the initial study, and then for the SVM B we ran more samples beyond the 800, also including chronic schizophrenia patients, to see can we validate it in the yet independent cohort, and this was the case. So you can see we can quite well classify patient population, but we also have a gray zone in the middle. So some patients we are not quite sure. And but it is very encouraging, and it also led to the pre-launch of this test in various US centers where it's also being reimbursed. The problem we have, it actually works very well. There's a bit of a problem in the US where people are very obese, and lots of these markers are changing as well. So this is a bit of a problem. But the main problem is that psychiatrists are not that interested to use that test. It's quite expensive, and they say, well, you know, if I see a schizophrenic patient, I can diagnose it very well. And they always say it's the colleague sort of down the corridor who needs to use the test because they're less good than I am. So, so there is some resistance of uptake, and this is what we are needing to address. So the field of psychiatry is probably not yet quite ready to embrace sort of molecules. We are not used to taking blood like, you know, people in the cancer field or diabetes, etc., etc. So there is a need for change, but we are continuing sort of on this project, and the centers who quite like to use this is also centers for drug-induced psychosis, or, you know, where they see drug patients, and they only have 5-10 minutes to talk to the patient in the US. And then they're either discharged or they may see a psychiatrist. They use this as a sort of stratification tool and triage tool. So we have taken that off the market now because we're wanting to improve it. And this is where we want to predict prodromal patients and then other products. And I talked to Marion about that, is really identifying treatment-resistant depression. So we did some market research. What do psychiatrists actually want? And obviously they don't want to diagnose schizophrenia so much. They want to identify treatment-resistant depression. Also the pharma industry wants to do that. Then also they want to predict drug response in MDD, much more in MDD. And so we have some markers for that as well, as I mentioned. And we want to also predict someone who comes in with a first presentation of depression, is this going to be a bipolar outcome, so that we can prevent pushing someone into a manic episode. It's the really holy grail. So these are products that we are working on. We have large samples sort of coming in, which hopefully will help to address this question. But there would be great opportunities maybe to collaborate with some of you, sort of to, you know, to get more samples to do these studies. It requires really a multi-center joint effort to validate such results. So the next one is another study. Again, this was a collaboration between the company, who are funding obviously the assays, and my academic lab. And a collaboration as well with Utrecht, with University of Utrecht, René Kahn and others. I think he's going to talk here in the forum, or has already spoken. So they have done this group study, which is a large multi-center Dutch study where they look at schizophrenia patients and also their first-degree relatives. And for about half of these patients, only half, I think, they've collected serum as well, and we run them on our platform. And so again, the question, can we, so some of these patients, so it was 120 schizophrenia patients and similar number of matched controls, and about 85 and 120 siblings of these schizophrenia patients, and 85 control patients as well, and they were run. So can we see an abnormality in the relatives of people who have schizophrenia? So this was really the question. And these are just here, again, the number 112. 16 of the patients, you know, the relatives are actually symptomatic. None of them actually have schizophrenia, mainly mood disorders, and 117 are asymptomatic and 87 controls. So these are just the demographics. Again, we used our platform, in this case with 190 analytes, I think, at that stage. And next one, please. So, and what we found is that the patients again showed changes which we previously found in our 51-plex, etc., and schizophrenia, particularly in the insulin, etc., but also the symptomatic and asymptomatic patients show highly significant changes, particularly in the insulin-related pathway, also in some inflammatory markers already. So is this an endophenotype? Is this a predisposition? I guess it is. I will tell you, if I have time, a little bit my theory about that. And in the end is our previous study where we already reported in this first episode studies these marker changes. And these are obviously treated patients, and the other ones were untreated. So it doesn't seem to be related just to antipsychotic drug treatment. And it is present in the symptomatic siblings who have depression will not be on antipsychotics either. And also the asymptomatics who are no medication. So particularly the insulin, the C-peptide, you know the C-peptide is actually part of the pro-insulin, so it comes from the same molecule, so it makes a lot of sense. The pro-insulin, the intact, so it's really the whole insulin synthesizing machinery seem to be affected. And this is very, very strong evidence. This can't be by chance. And also growth hormone, and I'm going to talk about that. Growth hormone is inversely correlated with insulin, the secretion. So what we see is in the relatives and in the affected and unaffected siblings, really these changes I just outlined already, but also immune response changes. So these two things, but particularly the metabolic abnormalities are present in unaffected siblings. Here again, just more details about the comparison, because these were drug-treated patients with the first episode patients. And again, you can see here insulin growth hormone ratio very severely affected. This is just making the same point again. And this just shows you the levels of insulin which are higher in here schizophrenia, symptomatic siblings and asymptomatic siblings, and the growth hormone goes the opposite way. So the ratio becomes quite apparent, and the next slide shows you these ratios. So if you normalize the ratio to the median, you can see here quite dramatically how with the presentation of the illness, this growth hormone insulin ratio is massively changing. Next one. So this is just a summary, really. This is further proof, and we found that already in the post-mortem brain tissue. We found it in cerebrospinal fluid, and we find it in serum of affected patients and also their first-degree relatives. We see changes in energy, metabolism, glucoregulation. So my hypothesis really is, and other people I think as well, so it's been sort of summarized in the selfish brain as well. The brain is about 5–6% of the body weight, but it uses about 70% of the body's glucose, this little organ. But the brain is the only area in the body where glucose has to be transported actively across the blood-brain barrier. So if there's an increased energy demand in the brain due to drug use, due to genetic predisposition, due to infection, due to perinatal abnormalities, brain damage, etc., etc. Under stress situation, this brain may not be supplied with enough glucose. And if you don't have enough glucose in your brain, this is equivalent to hypoxia, because the brain can't use alternate energy sources, as I alluded to early on. It can't use lipids, etc., ketones. So a reduction in glucose will drive down the glycolysis and the synthesis of ATP, and this will be equivalent to being hypoxia. And this is what we saw in the post-mortem brain, evidence of episodes of hypoxia. So we found hypoxia-inducible factors increased, glutathione, redoxon, so various antioxidant sort of pathways. It was not only pro-oxidative but also antioxidative. So there seems to be a system which is quite frequently fighting this sort of abnormality. The other thing, when we did metabolomics, so when we looked at the metabolites in the brain, mashed it up and put it through a NMR spectroscopy, we found the lactate levels increased in the brain. And it was reduced in the CSF. The glucose levels were reduced in the brain and increased in the CSF. So it is possible that somehow the glucose can't be taken up into the brain, because this is an ATP-dependent process. So you get increased utilization of lactate, which we also see in stroke patients, because when neurons don't get glucose, the astrocytes will provide them with some lactate to keep them going. This leads to a pH drop, and this leads to protein changes, activity changes, etc., phosphorylation changes. So this has a sort of knock-on effect, and maybe this is going on in psychosis. And this would fit with the hypofrontality hypothesis, sort of not hypothesis, that's been proven for 20 years, hasn't it? What is it else? That just there's not enough glucose in the frontal cortex, and this is what this is saying. So I think to think a little bit more simplistic, maybe schizophrenia is not that complicated. Maybe it's just not having enough energy. It's like pulling the plug out of the computer and then things go wrong. Surprise, surprise. So maybe nature is sometimes not these different regions and connectivity, but it may not be as complicated. This is a provocative little statement, but I just want to throw it into the audience. Next, please. So, again, coming back to this paradigm, is schizophrenia a systemic disorder? Does the brain and the periphery cooperate? Do they work together? And I think we don't question that. We had a fantastic lunch, and our leptins are being produced, feeding back, and now I had enough, but maybe I have a little bit more of this cake, because in the little sort of French little biscuits, which are fantastic. So, and then here the fight and flight system as well, so we accept these systemic sort of interactions in many aspects of our physiology. Why not in our behavior? Just because the readouts are slightly different. And I think this is maybe something to reconsider. These are the pathways. I don't go into details, not that I know them, but this is the insulin signaling pathways, and then in comes the growth hormone as well. So if insulin is increased, growth hormone is usually reduced because they're counteracting each other, and that is actually already linked with abnormalities in feeding and also in sleep effects, and on all psychiatric disorders, particularly also schizophrenia, we get abnormalities in their sleep patterns. So, these are all indications. It's not proven by any chance, but we need to put this into a concept, I think. Next one, please. And again, this is just really a summary. We can skip that and maybe come to discussion. So, conclusions about the last part. So when we look at first episode patients, we see a lot of metabolic and inflammatory changes, also in the more chronic patients. In the relatives of the schizophrenia patients, we mainly see metabolic changes, much less inflammation changes. So somehow I believe, and having looked at many data sets, but not really being statistically able probably to prove it as yet, I believe we have these metabolic changes as a predisposition. Again, the brain being so greedy, prioritizing and wanting so much glucose. And then in schizophrenia patients and their relatives, there may be genetic abnormality, which means that they haven't got maybe in the glucose transport, they're not quite as flexible and able to rev up the glucose supply under stress situations. So because we see the onset of the illness also in severe stress situation, biological, psychological stress, etc. But once someone gets ill, then we suddenly see quite a strong inflammatory component as well. And this is actually also what we see in diabetes. We see initially the glucoregulatory, then you get the inflammation because the hyperglycosylation of proteins changes in these pathways. They result then in inflammatory processes to be switched on. It's not fully understood, but somehow I think this is a at least two-step process in schizophrenia. So I would be really interested to undertake clinical trials to increase the insulin sensitivity in patients early on. like giving them metformin, other sort of insulin sensitizing drugs, but only those who have a strong metabolic alteration, maybe do a glucose tolerance test. So this is what we could take into the clinic. We would take a blood test, see this patient has profound insulin abnormalities, glucoregulatory abnormalities, glucose test is already abnormal, let's put him on metformin. You know, this is a normal prescribed sort of drug, and see whether this is improving outcome. Metformin may not be the best drug, and this is where one has to look because the CNS, you know, not all of these drugs go into the brain. One drug which is actually very good is pioglitazone, but it was taken off the market, which was also actually used for Alzheimer's disease, because these metabolic abnormalities, we see them in Alzheimer's and Parkinson's disease, and they also have a much higher rate of diabetes. So there are common pattern. I don't want to make it too broad, but there is a method in the madness, so to say, and I think we need to sort of look outside our field and outside of the brain as well, or it is worth it. You know, this is what I believe, and I hope I was able to convince you to also maybe have an open mind about it in the first instance. And well, we still have to prove ourselves as well, but it's something worth considering. Thank you very much.